I love energy because energy allows me to think and to act. Without energy, I would not be able to spend quality time with my family, and my work would definitely suffer. Today, I want to explain how the food that we eat plays a very important role in the energy production within our own bodies. Let's start with sugar. As you can see, the molecular structures of sucrose and glucose, which are two different forms of sugar, are depicted. Glucose is a monosaccharide, and monosaccharides represent the simplest form of sugar. Sucrose is a disaccharide and is formed by a glycosidic linkage between two monosaccharides. A glycosidic linkage is simply a bond between a sugar molecule and another molecule. A more complex form of sugar is called a carbohydrate. Carbohydrates are often stored in the body as glycogen, and various enzymes help transform stored carbohydrates into glucose. Dietary fats are not stored as glycogen. Instead, dietary fats are organized into triglycerides. All triglycerides have a glycerol backbone and three fatty acids, but triglycerides come in all shapes and sizes. A triglyceride may have three saturated tails or three monounsaturated tails. Maybe one tail is polyunsaturated while the other two are saturated. The structure of triglycerides within our bodies are dependent upon various factors such as diet, stress, enzyme availability, hormones, and other factors. Lipolysis, lipo meaning fat, and lysis meaning death, is basically the death of fat. Triglycerides are stored within fat cells, and we call these cells adipocytes. These cells are the site of lipolysis, and lipolysis is controlled by levels of epinephrine, glucagon, and insulin within our bloodstream. When epinephrine is high and insulin is low, the cyclic AMP pathway is triggered. This allows a protein kinase to activate. Simply put, a protein kinase is an enzyme that changes the shape of a protein, and this is done through a process known as phosphorylation. When the protein changes shape, another enzyme known as hormone-sensitive lipase is activated, and this stimulates the fatty acid tails to cleave away from their glycerol backbone. Once inside the blood, the fatty acid tails bind to albumin. Albumin is called a transport protein and transfers fatty acids to receiving cells. Fatty acids cross the cell membrane, and ATP, known as adenosine triphosphate, enables fatty acids to become acyl coenzyme A. Acyl CoA is metabolized into acylcarnitine, and acylcarnitine is able to cross the mitochondrial membrane. Once inside the mitochondria, acylcarnitine is metabolized into acetyl-CoA. Glucose enters the mitochondria through a different mechanism. First, insulin within our blood binds to a G-coupled receptor on the cell membrane. This stimulates multiple metabolic functions, which eventually causes the GLUT4 vesicle to bind to the cell membrane. Glucose then enters the cell and is eventually metabolized into pyruvate, and this is done through a process known as glycolysis. Pyruvate enters the mitochondria and is metabolized into acetyl-CoA. I know this is a lot of information to take in, so I extracted a few key points and posted them for you all to see. Even though fatty acids and glucose have different ways of entering our cells, both molecules create acetyl-CoA, and acetyl-CoA is an extremely important molecule when it comes to energy production. Acetyl-CoA is important because this molecule enables our cells to create ATP, and ATP is the energy currency for our cells. As you can see, glucose produces approximately 34 ATP, but palmitic acid produces a whopping 131 ATP. Even though glucose always produces the same amount of ATP when metabolized, ATP production from fatty acids varies and is dependent on carbon chain length. A fatty acid can be designated as short, medium, or long, and longer fatty acid chains create more ATP than shorter ones. Now, you may be asking, how does this work? Well, let me explain. Acetyl-CoA is labeled in pink. Acetyl-CoA enters the citric acid cycle and is metabolized into various substrates, such as citrate, succinate, and malate. This cycle requires various cofactors, such as adenosine diphosphate, NAD+, FAD, and water, in order for one complete turn in the cycle to occur. Well, these cofactors are metabolized into NADH, FADH2, ATP, and carbon dioxide. NADH and FADH2 both produce ATP. Specifically, one NADH is equivalent to 3 ATP, one FADH2 is equivalent to 2 ATP, and thus one acetyl-CoA is equal to 12 adenosine triphosphate molecules. On the right, palmitic acid is depicted. This free fatty acid, which was once part of a triglyceride, can donate 16 carbon atoms to the citric acid cycle. The carbon on the left side of the structure, is part of a functional group, more specifically, a carboxyl group. The carbon from this carboxyl group will not be used in the citric acid cycle. On the bottom right, an acetyl-CoA molecule is shown, and you can see that this molecule has 2 carbon atoms. Well, palmitic acid has 16 carbon atoms and can create 8 acetyl-CoA molecules. In order to cleave 2 carbon atoms from palmitic acid, another process has to occur, and this process is known as beta oxidation. Palmitic acid must undergo 7 rounds of beta oxidation in order to be completely metabolized. Each round creates 1 molecule of NADH and 1 molecule of FADH. When we add up the numbers from beta oxidation, the process produces 35 ATP. When we calculate ATP production from the citric acid cycle, we know that 8 acetyl-CoAs must be metabolized. Because each acetyl-CoA is equivalent to 12 ATP, a little math work would help us calculate 96 ATP.